Okay, so our next speaker is a real pleasure for me to introduce. Piet Gros will talk about the complement system. And I personally had the pleasure of working in Piet's group in Utrecht for several years as a postdoc, and it was a wonderful place to be where there were people doing great molecular biology in the lab and also people trying to develop algorithms in the computer room, and it was a lovely place to be. And whilst I was there, Piet won the Spinoza Prize in Holland, which is the highest scientific prize you can win, and it's named after the Dutch philosopher from the 1600s. And one of the things that Spinoza said, and I have to get this right, he said that all things excellent are as difficult as they are rare, and I think that sort of resonates with people chasing hard and exciting biological targets in the lab.
Say that again. What was it?
Did they not check this when you won the prize?
No,
I thought you have to know. So all things excellent are as difficult as they are rare.
Okay.
So with that, Piet, I hope your talk will be, well, excellent.
So I hope to give a rare talk then, which doesn't sound right. Anyway, I'll do my best. It's been a great pleasure to be here. Thank you for the invitation to come here. And it actually makes me feel like a PhD student again, because it's the EM stuff. It's like an academic candy store. Yeah, so there's so much new opportunities, new questions that we can address. So it's a lot of fun. It's very inspiring to see this big revolution in the whole thing. So I find it a lot of fun. So this was to be rotating. Did it rotate? Anyway. So let's go. So what I'll talk about is actually on the complement system, what we worked on for 15 years or so by now. So a lot of crystallography to start off with, and we're now moving into EM, and I'll show you, I think, what I find very fascinating, some tomography stuff, to see these molecules in action on a surface, because that's where it happens. And at least it's tantalizing. It brings pictures, which perhaps you can't call science yet, but it's very intriguing, and I'll show you that at the very end, I hope. Okay, so complement system. So this is part of the immune system in your blood, basically, mostly. And it's not dealt with very much in textbooks. But this is basically an archaic defense mechanism, which is already arose at multicellular early species. It's partly still present in flies, in fish, and it's still present in our body. So it's there for a long time, much older than antibodies, so that's just a new kid on the block. And basically what it does in our system, still in our blood, and also in the fluids between cells, so in tissue, it can recognize and eliminate bacteria, viruses, but also altered host cells. And of course, this is just imagine. Of course, it's easy to imagine that you can clear bacteria and viruses. They're quite alien to us. But you also have to clear your own garbage, and that's of course very tricky. And you have to do that while protecting your healthy host cells. And that's being done by this system, and actually it works in a very archaic, simple manner. Actually, the question to that is very easy. So for structural biology, of course, what drove me to this topic. It's a big system of many proteins, multi-domain proteins, so there's a lot of structural rearrangement going on. So it should be fun. Now, if you go to a hospital when you have some disease, hopefully not as your newborn kid yet with this terrible stuff, but you don't find you don't find this board and say, well, this is where you find your complement doctor. Still, it's now actually also in complement there's a revolution going on. Because besides just you being like for infections, and if you have genetic effects, then it actually causes basically problems early life in kidneys, or very slowly in the eyes. And so the most common form of elderly blindness is related to slowly building up of garbage in your eyes, and it just affects the cells around it. And also if you have like chronic disorder or inflammation, if you just disturb your tissue for a long time, it's for example in a stroke or a heart attack, things change, and then complement comes around and starts to clear up and cause you big damage. So that's kind of all classic. Also periodontitis for the elderly people, for the older people in life, Yeah, you have this horrible cleaning of your teeth. And it's basically, there is a compound out there which, so hopefully within a few years you have competent toothpaste and everything is gone. Anyhow, so also in CNS, rightly, the last couple of years, it's become clear that also in the brain, complement is important, even though, of course, this is locally produced complement. So Alzheimer, so that's in terms of functional activity, it's like very similar to the drusen that it builds up in your eye. So what happens is that you get this buildup of the plaques, and you get complement responding to it, overreacts, can't clear it, and then because of the overreaction, it kind of clears the neighboring cells, and that's where you get your loss of memory from. So that's kind of still an archaic overreaction effect, but also in synaptic pruning. So somehow, so if you have badly working complement proteins, you have an enormously high chance of getting schizophrenia. So somehow these complement proteins figure out which synapses to prune and which not. So so the whole picture that comes out of it is it's much more a very general function of tissue surveillance and maintenance. So there's possibly a much more tricky, subtle way of how this thing works in keeping your tissues in a healthy state. So this excites a lot of people, and this makes complement really a lot of fun to work in at the moment. Now, 15 years ago, 16 years ago, we just stepped into this because it's a nice network of large modular proteins. Lots of complexes are being formed and cleaved. And altogether, all these proteins, they cause a rapid and localized response in clearing of the garbage somewhere in your body. So it has everything you, of course, want in regulation. It has to initiate suddenly at a certain place, amplify, and of course you got to stop it in time again, and of course something has to happen as an effect of that. So these are the major proteins colored by homology. So the backbone is these three up here, C3, C4, C5, illustrious names. Proteases in red here, and that's kind of the basic system. Then there's like initiation factors, specific ones built in front. And there's these protection factors of protecting healthy host cells, kind of controlling all of this. So how does this work? So just to make, I can give you the schemes from the textbooks, and there's a lot of protein numbers and so on. So let's just look at the surface of what's basically going on. It's like, okay, you have different mechanisms to start this off, either specifically, for example, through antibodies or binding to glycans, or something which is not quite understood, it's just a low level of automatic turnover. That also actually converges in the cleavage of this C3 protein, which you have like in 1.5 grams on average per liter in your blood. So you're basically, each of us are fantastic overproducers of this C3 molecule. So this is then, this is the resting agent. It's activated by cleaving off this little domain. That exposes thioester, and that just binds somewhere basically randomly to any surface. So then two proteases come around, factor B and factor D, and they just create on top of this platform an enzyme complex that cleaves more C3. So then it's like localized on the surface, local amplification, and you get this enormous spread suddenly. Yeah, so that's called in immunology, that's called opsonization. But it's basically producing a lot of these molecules that just within a minute or so cover the whole surface. Then another protease comes around and makes other fragments, and it sees these different fragments that actually have different responses to like phagocytosis macrophages, taking up the macrophages or stimulating T cells and B cells. And then the third possible route is actually homologous to these two again, C5. It's cleaved in a similar mechanism, and that picks up several other proteins, and you get these big pores of 100 angstrom. This is the membrane attack complex. Now this is all going on, and particularly this one is aspecific. Yeah, so this happens everywhere at a low level. And you need these regulators, as they are called. So on the surface or in the solution, they actually recognize your own surface back again. You have these proteins that just keep this step, the amplification, under control so that you don't ruin your perfect cells. Yeah, so this is kind of the way it works to protect your healthy host cells is basically a balance between attack and protection. As long as you send out this thing that attacks everybody, and as long as you can protect your own cells, you're fine. Yeah, so it's a very archaic immune system that doesn't recognize anything because you just protect your own cells. So the healthy cells can protect themselves, and otherwise you're lost. Yep, okay. So what I'm going to tell you today is first, give you a little wrap-up of the old work, summarized 7 years in 3 minutes of movie. Then continue, the first part I will continue with how this regulation works at this level and how these different products are produced to make this stuff. So the movie of 3.5 minutes, 7 years of work. So this is C3, which is 16,500 residues, 13 domains. So in those days, that was really a rather big protein. Now with the EM people, this is all tiny. Yeah, so the little domain is cleaved off. You get a conformational change. This is 85 angstrom distance, so it's a huge conformational change. We were only used to like a loops moving like 3 or 5 angstroms. It was exciting. So this was 85. Local rearrangements, making the thioester in an acyl imidazole, and that responds, that binds to any hydroxyl on the surface. So sugars or proteins, so anything. So the first step is then that protease factor B, which is inactive in solution in plasma, that swims around. And why it's actually inactive? Because the cleavage site that has to be cleaved to make it active is buried. Yeah, so the first arginine is pointing in, that has to come out before it can be cleaved. This binds to this C3b molecule, this platform on the surface. That's a whole tricky process, and it also needs a magnesium ion. This is like an integrin A-like type domain, and this is the minus side with the magnesium ion, and that induces a conformational change that makes this a dynamic equilibrium flip-flop, and that is associated with actually making this loop accessible for proteolysis. Oops, there it goes. Of course, there's nothing, this is just flopping around, so there was no density, but just sitting somewhere out there. In solution, this is then recognized, this new shape is recognized by the next component, which is called the factor D, which is inactive by itself, and this is a very specific one. It's activated by this one substrate that it has in its whole life. Yeah, so it's been activated by the protease factor B, activates its own enzyme, and then this is all fantasy, how this goes in, because we don't see that. Yeah, and it's cleaved the factor B for activation, and so this is also the conformational change to turn on D itself. Yeah, so it's a very strict cleavage process. Only this one substrate is being done because it can activate it. And then magically, we don't know how that works, actually this all falls off. And then finally, you have this final swing, which is the enzyme that amplifies it. So this is we call it C3 convertase. This is the amplification enzyme. It binds a new C3, cleaves off the little red domain, and then probably in kind of this orientation, directs this directly to the surface, and that just keeps on going rapidly, and that just fills up the whole surface. Of course, each of these can be a starting point for a new protease enzyme. So this is like amplification, as you would expect. This is rapidly going, and this goes on until you have one of these protection factors that kind of just break up this complex, this non-covalent complex. So that's 7 years of work. I think I'm very grateful to these 4 people that did all this spectacular work. It's fantastic it's been great fun to work with them. And a special thanks to Federico, not just because he's in the audience, but he also is responsible for actually making this final movie. And it was a big job, but very rewarding. It's a very nice movie. Okay, so why does nature do this? Right, so this is the choreography, summarized, all of these different structures that we solved. And I think the answer in that is actually hidden at the last step. The enzyme, the convertase enzyme, has a half-lifetime of around 90 seconds, which is actually quite long, and then it dissociates irreversibly. It doesn't reassociate anymore at this level. So it's kind of an inherent turn-off signal. And you have to start all the way from the front again to get this going. So why is that? That turns out to be purely kinetics. Yeah, if you look at structures that we have, and of course we don't have everything at the resolution we would like, but there is no signal, no indication that there's a structural reason. Now, doing SPR, we could show actually that the binding has two-step binding process, has a fast and a slow rate, And the slow kinetics is completely magnesium dependent. Yeah, so we can also do with protein fragments, but also, of course, with ETDA and the BA fragment only. So we have a fast association and a slow dissociation. So what this basically means is that the propeptide, which is these yellow domains, is actually the fast on and off rate. So that kind of sticks it on, and that just creates time to actually get this magnesium in and hook up this final thing here. And that then also means that it stays on for a long time. So you have this 90 seconds complex that actually covers the surface. And then it falls off and it never comes back. Yeah, so this is why nature has basically this loading complex. So now going on, this is also where the regulation happens. Yeah, so you have these regulators which all have CCP domains. Yeah, so they're not CCP-EM or CCP4. There is a CCP4, but the complement control protein, that's obviously what CCP stands for. Yeah, so there's the same domains as the yellow ones, but all the regulators have like either 4 or up to 20 or 30 of these domains consecutively. And they can actually break up this complex, and they also help recruit cofactor, this factor I, this next protease, and help in dissociating and cleaving this thing, which is the inactive species, which cannot be used to make new complexes. And it's either 2 times or 3 times. That has a difference in either stimulating B cells or T cells or phagocytosis. So the question that we then, of course, asked is, so how is this recruitment working, and can we explain why the activity is different? So already in the movie you saw one structure which is done, this is factor H complex up here with 4 of these domains. Basically it's binding with only 7 micromolars, so it's very weak. And obviously that's also one of the things that's very hard to do with EM, these weak complexes. A huge binding interface and only 7 micromolars. And this is actually critical for regulation because all of the mutants that you find in these kidney diseases kind of map in these regions. So how does this work with factor I? Piet Gros is in the audience. He did the structure of factor I by itself when he was in Susan Lee's lab here in Oxford. A great job. It was a very tricky crystal structure, so it's a very impressive piece of work. And this is the C3b with factor H. And obviously, it actually has to cleave 3 times in this blue domain. So what you would expect is, of course, that this thing with the enzyme, the catalytic domain, just docks on these binding sites up here. And there's this nice groove, so that's probably where it sits. That explains that if you cleave this one, you actually break down the factor B binding site. So the first cleavage stops the whole amplification process. That's quite easily explained. Now next. So there are many different regulators, or several, with all different activities. So part of the activities I don't even show, where there's this different pathways. But also here, so this is the component which only binds, and basically it's actually the whole construct is here, but this is kind of completely floppy. So this binds at the bottom, this binds at the top, and different affinities and different activities, like there's breaking up, which is called decay acceleration, or the factor I stop, which is called cofactor activity. So that's also shown here in the binding patterns. Yeah, so this is bound here at the bottom. This actually here is actually, if you look at the density, this is very weak density, so this is very poorly contributing, so it's mostly binding up here. And this one that can actually cleave 3 times, this one, it's actually sufficient for cleaving 3 times. And most of the other ones only do 2 times, or this one doesn't even cleave anything. So I just put this picture in this morning, because this is like crystallography crystal structures, and it just looks like an EM structure, right? So lots of structure variation, but this is basically a collection of different structures. We know the flexibility, this just jumps up and down, and this moves all over in the crystal structure. And then the question is, how do these different regulators affect different, so either cleaving 2 times or 3 times, or not at all? So the crystal structure was done by Xiaoguang last year in the lab. So this is a pretty low resolution for us, of course, 4 angstrom. But it's all pretty much, this is a factor H, and this is where the factor I sits. It actually sits up here, not quite as expected, because it actually shifts around this top domain. This head domain is actually twisted around, and it contacts the backside of this domain. which is very interesting to us because there was one mutant, set of mutants in factor I or in C3b which you could not explain in previous data, and that is now used completely. Where is it? This one is a complicated picture, I must admit. But this is basically the residue that was not interacting with any other protein, and it's now in factor I, so that explains why that's a disease-related mutation. So all of these, basically this whole binding set at the top fits with biological data that this is really critical. Now, the major question for the field is how is factor H on the regulator used? So the interaction is completely mediated by these two 3 domains. Showing up here hydrophobic interface with a charge in the middle. That shows directly why this DAF enzyme, the homologous DAF, does not do it because it doesn't have these hydrophobic patches, and that's then already fits completely with the data done in India by Sahu's lab. And then basically if you look at the enzyme, so this is the interaction, and the major thing that happens to factor I is actually that in the structure that Piet Gros did in blue, this is a free structure, so a major part of the enzyme was not structured, and now it's completely structured. So what happens is these red loops are completely structured. The contacts with factor H are critical to actually form the enzyme to do its job. So it's like thrombin, but it's been shown that the activation is that the interactions work through stabilizing the fold, and that turns on the enzyme. So if you don't stabilize it, it's a very poor enzyme. And so that's also how factor I works. It's just completely stabilized. And so it's only active in the complex due to this stabilization. Yeah, so for the first cleavage site, everything is quite clear. So it's docked directly on the first cleavage site. So it works through this contact is required from the top. So all of this helps to make this compact shape which stabilizes the structure. And that turns on the activity. Obviously, we had a serine-to-alanine mutant to stop it, of course, to capture it in the act. Otherwise, we couldn't do this. And that just induces the stability induces the activity, and then that cleaves this loop, and that loop also is more flexible, etc., etc. So how is the second and third side cleaved? Well, that's a bit more tricky. So we actually used one of the other the viral things called PCP to purify more or less the species which only cleaved once. So that's shown up here. So we have a more or less pure, some major fraction of C3b1 and some C3b2. And we did negative stain and Sachs. And basically what comes out of it is that the C3b structure, one cleavage of C3b, does not affect the overall structure. And you need two cleavages, and then the whole thing falls apart, and the thing becomes extended for the third cleavage. So how is that related with activity? That's more or less we try to show it here. And so these are the 3 major ones that do this job. So these two cleave only basically 2 times, and this one cleaves 3 times. So this here in the crystal structure is actually, the rest is here, but just floppy. So this one depends completely on the interaction up here. And you can see that if you cleave the first time, once you start cleaving, actually the activity is completely, the binding of the regulator is completely lost. So it's the regulator binding that actually determines the activity. So when you don't have at the cleaved species, the regulator falls off, and therefore you don't have activity anymore. Whereas CR1, which binds completely at the top side, is completely independent of the cleavage and stays on. So what you have in this particular case, so you have the enzyme complex, it's completely stabilized, and this is a very compact arrangement. It's very hard to imagine this could move. So what we think is actually this stays in place. You can reason that if you have one cleavage, nothing happens, and the second cleavage site is just a couple of residues down the chain, so that can just flop around and dock into the second site. Then if you have 2 cleavages, this thing starts to unfold completely, this domain, and then the third cleavage is actually at the top end, so that could go into here as well. So that it's this, after 2 cleavages you get this structural change, but then if you're completely dependent on this interaction at this bottom level, then you fall off if you don't have this interaction. Yeah, and only the regulators that depend on the top ones only, that bind stronger up here, stay on and can do 3 cleavages. So that's trying to show here. It's a bit complex perhaps. Yeah, so we have this different binding patterns, some bind more at the top, some bind more at the bottom. So it means if you depend on this arrangement to bind, then you can only cleave 2 times, because if this binding site falls apart, then you just fall off and you don't cleave, and if you don't support the cleavage further. Otherwise, if you bind mostly at the top side, then the whole thing just keeps on going, and you get finally the last product, which is this little domain only without the thioester, and that's very powerful in B-cell stimulation. Now, to link that to biological function and immunology, it's a bit too short to say that this is directly explains everything, because biology is far more complex than structure biology. Okay, so now let's move to EM, because actually I think you were not waiting for complement, you just basically want to see EM structures. So what happens is actually we have this process of C3b, and so the movie that I showed you, this choreography, making this enzyme complex that's duplicated in life with C4 and C2. So basically you have the same like C3 and factor B, this is duplicated up here. And now it's not cleaved by factor D, but now it's cleaved. So it says both steps, the cleavage of this one and the cleavage of the enzyme, are done by these big complexes. And not by this, the second step is not done by this soluble form. Yeah, so you have the antibodies, for example, that we worked on, a step here, and then it turns on and sets up the whole system. So it's kind of duplication. So this is duplicated in evolution to just allow this new hookup up here. Now the antibodies, of course, are quite important if you want to apply that also to tumor therapies, clearing up tumors. That, of course, is done a lot already using Fc receptors, but it also can be shown that, and actually complement plays a major role in this, and certain antibodies are very powerful in doing this. However, it's totally unclear what actually determines the structure of antibodies that activate this first complex. Yeah, so now we get into the EM region, because this is 1 megadalton, and so it starts to add up to have some size. But we don't know, in solution, we cannot reproduce this, because we don't even know what's going on. We don't even know how many and what this shape is. So there are very few complete IgG structures in the PDB. I believe there's only 2 or 3 or so. This is one done from HIV. Yeah, you have the Fabs and you have the FC. And this is what actually, the model is a theoretical model, but it's pretty much proper. I think it's very good. So C1q has these recognition units, and it's all like a bundle together by collagen. So there's like, they describe it as a bunch of tulips. And well, clearly they're not from Holland, because it doesn't look like tulips. And then these proteases sit there in the middle somehow. And that activates, the model is that when this docks, there's clearly a surface trigger when this docks. This somehow is activated. These are proenzymes that become then activated into complete enzymes. So the cleavage is then happening. And then actually you can cleave C4. And the cleaved one, so this is C4b, like C3b. And then it binds the protease fragment, similar to factor B. And then this one, this complex has to do the second cleavage as well. So it's a concerted. That's one of the big questions, and I'm not going to give you the answer because we don't have the answer. So we started off to get some insight in this with tomography, collaborating with Bram Kosten. I'm very grateful to Bram and Roman in Leiden. So this is just collaboration. It also predates Nielsen, so that's not the classic range. This is just like how people work together. So what we did actually, this is done by Christoph Dieboldter, after some trials which I offered, which didn't work. He found out himself there is actually a whole system that you can simply buy, and it works straight off. Yeah, so you buy these vesicles, which are used in hospitals to measure complement activity. You add antibodies, you throw in blood, and the whole thing is just built up, and in the end you get pores, and then just the chromophores are released, so the self-quenching chromophores, fluorescent molecules, and you can just measure the activity. Yeah, so this is all developed, and if you stick this in the EM microscope, it straight off works. So instead of trying to develop our own system, which he tried for 1.5 years, following my suggestions, and we gave up, and then he just started the system, and it works. So what you see here is sort of vesicles by themselves, of course, are completely, this is like inside vesicle, but it's just if there's no antibody binding, it's completely naked. And here you see one layer of antibodies. Yeah, like that. What is it? 11 nanometers fits with one layer of antibodies. And all of this mush around is what's added by complement. if you had normal human serum. Now, if you only just do the first step, you bind C1, you have this, what we call, for a long time, Eiffel Towers. Yeah, clearly they're Eiffel Towers. And you have these, and these ones are the C1 complexes bound to antibodies. So this level here fits exactly with the level of this first platform, and on top of that you have the C1. And so obviously I wanted to have the structure of that. So what Christoph did is actually do subtomograph averaging using Piet. And in the end we had 100 particles and got this meager resolution of 6.5 nanometers. If I would have known that in advance, I would never have started it. But we were very lucky in a sense, because this one structure that I showed you, this antibody, intact antibody against HIV, actually crystallizes in B6, at least it has 6-fold axis. And if you look at that, it actually has this ring of FC fragments up here, and then at the edge it has one fab down and one fab in the ring. Yeah, so it's up and down, 3 down, 3 in the ring, and 3 up. And we have this density at 65 angstrom resolution. And if you just take this structure and you flip around at the 2-fold axis these structures, you make one ring appear with all 1 fab down and 1 fab in the ring, and it just perfectly fits this density. Yeah, so that was for us a suggestion that this 6-fold would be one of the platforms that is recognized by the 6-fold C1 for activation. Yeah, so structure biology, there's no backing up. Anyhow, so we were very lucky because this was that our collaborators down the road were actually working on a similar problem. One of actually the leader of the team actually is one of the co-authors of the antibody structure that we used. So the idea actually was around longer without us knowing about it. So this is the vinyl structure. And then, so using with the antibody company, what they actually figured out is that at the FC interface, you can actually have mutations that completely knock off activity, and you can even have mutations that activate, that stimulate, that get a stronger binding, and by having a stronger binding up here, you just get increased complement activity. So, and one of the very cute experiments I thought is that what they actually did is using this antibody model that we had, and using, so that's of course 2 fab arms of the same nature. They made a bispecific one, so a combination of one arm intact and the other arm just completely arbitrary, and it still works. So that actually fits with our model that it's 1 leg down and 1 leg up in the air. Even better, they use an antibody against EGFR, and that antibody has absolutely no complement activity, none whatsoever. Yeah, you make then a bispecific first, so 1 leg still intact and the other leg is just completely arbitrary. It's like an enormous, strong complement activator. Yeah, so the model that this actually gives is that you have 2 fab arms, they both bind strongly to the surface, the FC arm sticks up right in the air, and you never make this platform. Yeah, so then you make this 1 leg beast that actually creates a chance, the opportunity to make this hexamerization platform appear, and then you get very strong complement activity. So that was very nice, I thought. So this is now followed up by a postdoc that Bram and I had together using. So this was the tomography actually done previously. Was like one of the very first cryo machines in Eindhoven. Actually, so it's like a very old detector. And anyhow, it's like nothing compared all of the phrases that we throw around today. So then Tom Sharp actually used the Volta faceplate to actually look at the same process. And then also we used, like in this, do it step by step. That you can buy basically serum with single components deleted by purified out. So you can look at complements step by step. And you can see how this evolves. Like of course it doesn't, it just like until the end of the road each time, you just have it step by step. And that is fascinating. So what you see, for example, so this is faceplate. Now this is the antibody layers again. Here we have a whole group of C1 molecules. So this is just having C1 added. This is the antibody added, and C1 only, and no C4. So it stops right after C1. So these are just antibody complexes, very clear now. And this one is, I find amazing. So it's like the C1 occurs like with and without the protease platform. So there's a minor fraction that doesn't have the proteases bound. Yes, this is like this collagen thing, this big collagen bunch of tulips sitting up here. So what you look at here is a single molecule, and this is a collagen. Yeah, so it's one collagen what you see, 3 peptide chains. This is what you can see in tomography, and this is absolutely clearly C1q. Yeah, there's no doubt about that. So if you're lucky, you can see single collagen chains. Now there's another one where I'm less convinced of, and there's more bias because I would love to see it. As this next step, so we have actually, so the process here, the next step, compared to the process that I told you in the movie before, now we have this big complex which is bound to the surface, has to cleave the first molecule, C4 into C4b, then bind C2, and then cleave this. But it binds then, in the meantime, it binds with the thioester to the surface, and this is a fluid vesicle, so most of them will just walk away. Yeah, so it needs to happen. So there's many solutions possible. Of course, it could be multiple complexes, like squeezed between complexes, activating it. But what we see on the surface is actually the following. So here you have a C1 molecule doing nothing, like most of them. So all of the products have walked away. So there's lots of stuff here, which is probably C4b because there's nothing else, antibodies in C4b. And here it's just exactly what we expect. Yeah, this is one of the Eiffel Towers, and this is little density sticking out. That's like the protease domain sticking out, binding to the substrate, C4b sitting here, waiting for C2. Now this is not science. Yeah, this is N is 1. Yeah, so what we're now trying to do is actually use biology to actually prove this, to get it to the big numbers, and also show the next step that we can actually prove that this is happening. Of course, I believe this is happening, but you shouldn't. Yeah, so anyway, so you can look at the whole process. So this is when you have C5 depleted. So there's no pore formation because C5 is critical for the pore formation. And so what you end up is like the C1 complexes, which you can easily recognize. And then the rest is just covered by C3b and C4b. And we would love that the experts in the room do the job even better, because that we can actually look and count this 150 kilodalton protein and just look at statistically what's going on precisely. Now the other movie is like when you have kick out C4b, so what you expect then is actually pore formation through the classical pathway only. So it's like the C1 complex, which should be tightly linked to pore formation. And so what you see here is like you have these C1 complexes in the middle of this pore. The color is correlated with it. So blue is a very high correlation coefficient. And so we have these objects which are clearly, and then we even have, and that happens as well, where it's misfiring where the complex actually formed in solution. Yeah, so there's lots of fascinating stuff going on. And we don't know whether this actually means very much, that it's actually sitting and comes together. Yeah, so there's a lot of things, inspiring things for future research coming from these tomography data. Okay. Where am I? Okay, so the final thing that I'm going to talk about. And it's 35, so I'm actually doing quite on time. Yeah, so finally, so I spoke about this, and then the third pathway, which is actually also based on the C3, C4 molecules, is the C5. C5 is cleaved, probably very similar, and actually in a similar and related convertase complex. So somehow in the process, this substrate specificity of the enzyme complex here is changed from C3 to C5, and we have no clue how that happens. Well, we have suggestions, but no data to prove anything. So this is cleaved, making a similar, probably a similar species, but now it's slightly of different enzyme, with different protein. And that picks up C6 to C9, and these are soluble proteins, and they then penetrate the membrane and make a 100 angstrom pore. So all these have the MAC-PF domain, so MAC, or membrane tank complex, the complement, and perforin, so the perforin up here. So they have this central domain, which is responsible for the perforation, and all of these ancillary domains on the side, which is basically all of these things that are there for regulation. That's saying now we don't know. Anyhow, so we did the structure of the MAC-PF domain of one of these, and that's shown up here. And actually, this is actually similar to the bacterial CDCs. And that's been shown by Ron Threaten's group, that actually they work by these helical elements in the bacterial form swap around, making huge beta strands that make the pore. And it's also by Helen Sable's group, EM work shown to work that way. And structurally is that these beta strands extend and make the big beta barrel pore. And that's how complement would look like. So this is a structure that we did of the first step, which is C5b bind to C6. So this is 13 domains and 10 domains. So, and in the old days of crystallography, we would be proud of this. It's 23 domains, and all you guys in the audience go like, oh, I got 100, 200 domains, 400 domains. Anyway, so this is actually the cleavage process. So this is what I showed you before, the 85 angstroms movement. This is a similar thing happened with C5. Actually, it's captured halfway by this new protein. And that actually fits with the biological data that if you feed C5b6, the cleaved version, to C6, nothing happens. Yeah, so it makes sense that it's captured halfway, because if it's just extended and it's just dancing out here, it's apparently not active anymore. Now, together with, what should I point out? Because this was all, part of it, it comes back later actually as well. The EM work, we fit this also with EM work with Dorian Bubeck, who now has her own lab in London. And it's been fun to collaborate with her and also talk with her recent data that she showed here on her poster. So it's a lot of fun. So what we did next, Tom Sharp did next, is actually look at how vesicles, how the pore forms on vesicles. Not, in this case, not just using the whole complement process, because then it's all modeled up by different proteins. You don't quite know what you look at. So if you just start half just by the C5b6, then you start adding 7, 8, and 9, then you get also by itself pore formation. So this is only the first step. C7, you get this blooming on the surface, and no punctuation, and that's known biologically. And if you then add C8, you get a small pore already, and you don't need C9. That also fits the biological data. And then if you add C9, you get lots of variations. You get single pores, and you get all these complex structures that are out there. So Tom averaged this, did subtomography averaging, and he got this structure. So he averaged, just to point out, he averaged all of this, right? So no surprise that we have only, where's the number, around 20 angstrom resolution, because we have like many different complexes. This is the average structure, but still it shows you the big features, the C5b6 on top, and then the 6, 7, 8, and should be somewhere here, and then the ring is made by multiple copies of C9, and that makes this pore, and there's this big seam in here, this kind of smeary, like a poorly closed thing, and it's kind of slightly cone-shaped, because that fits with the length of these different. C6 is smaller, so you kind of put this below just to fix the length of C6, and it all makes sense. At the same time, Dorian actually, and also Helen Sable's group, I say Dorian because he had the whole neck complex, and Helen Sable, together with the Australians, did a similar structure of C9 only. So you can do the whole set in actually as a non-physiological complex, but of course you get a big picture of what it looks like on C9 only. And then this is one purified from vesicles with C5b6, 7, 8, and 9. These structures, and surprising thing here is that actually, as we did not expect, that all of these high-resolution things have exactly the same number of copies, protomers of C9 in a ring, which actually fit with what we estimated. And this is the superposition of the structure from us, out of the membranes, and this is the structure from single particles. And obviously in the paper we made a point in trying to say this is much more real than this, but to be honest, it's basically the same. Right? So, anyhow, it's been a lot of fun. I think the basic points here is that the fun thing as a structural biologist is to show, this is what I tell to students at home. I say, so what's molecular recognition in life? Well, they typically come up with the answer, it's affinity. And then you ask, like, what's molecular regulation? And basically, it's all conformational changes. Right? So I think we're, as in many different fields, we'll see these massive conformational rearranges, not just little loops, but basically also big things, like structures just turning around and just basically going from one state to the other state, and that's just conformation. Because you get a completely new phase, of course. If you just make conformational change, it's just making a drastic step in time in regulation. We also see quite often is actually that if you look at how this may have evolved and looking at data from people that study these things, there's bits and pieces are actually reproduced, copied, but nature just doesn't copy it perfectly. There's always a new twist to it. That's just making slight variations. Modification of the same scheme gives you new ways of control, and that just ends up in new regulatory features. Yeah, so that's kind of a recurring theme in our structures. So the IgGs, the major thing for immunology is that you can actually now direct complement on cells by enhancing FC-FC interactions, and that's very stimulating for that particular field. And then actually, and this is for us a tool to go on and look at it better. Amplification, we show how this process of conformation changes builds up the convertase complex and activates it. And turns out an active enzyme complex is completely I didn't mention that in detail, but basically the enzyme, so the whole C3 binding is completely done by this interface, which is like the dimer interface between C3 and C3b, so it's just a dimerization process that just drives the whole orientation. And then this whole business about the loading of the propeptide and cleaving off is kind of like building up a timer to have a long-living complex, which is only used once with a slow on and off rate, so that it dissociates irreversibly. Oh, I didn't mention this, but this is like a very nice molecule that's made by our long-standing collaborator inhibiting convertases, and that's in part that could end up in your toothpaste and doing much more important things than that as well. So here the protection and the recent work by Xiaoguang in the lab, the regulators all bind, and this was a very surprise to the experts in the field. They all bind in the same mode, and they all bind in different ways on top and bottom, and that determines their activity in helping factor I to work. And in MAC-POR, actually, basically that's where bacteria and mammals use the same mechanism. So that actually, my last slide, I'm very happy, and it's been fun to work with my group. So this is a picture that we had some years ago doing X-ray data collection. Some people here actually in this room. And I'm very grateful to collaborators and also to Bram Kosten and Roman Koning for giving the opportunity to start off this EM work. And in the meantime, we have hired Friedrich Förster in Utrecht, and this summer Thea Zeev Ben Mordechai from Oxford will come as well. And so we have our own machine and expertise now building up in Utrecht to do all the EM work at home and having more fun. So I thank you for your attention, and welcome.
Well, thank you, Pete, for an amazing talk, and it's wonderful to see biology at so many, on so many scales, I think. So, questions for Pete?
Too much data.
So,
Pietro.
First off, I want to say how wonderful it is to see answers to questions that a lot of people had for many, many years, and how elegant it's been elucidating all these mysteries. It's really wonderful. And then I have one question, which is related to this striking match between the symmetry of the C1q complex and the underlying symmetry of the FC hexamer. Because the first thing that comes to my mind is the lectin pathway is also activated by bunches of lectin-bearing molecules. And could you comment on the possibility that the hexameric nature of the molecules that activate the lectin pathway have to do with some hexameric epitope on the cell? And do we have to think of antibodies mediating this interaction between the lectin complement pathway and the sugars on the surface of the cell?
Commenting on that question is, of course, easy. No, I think it's different. I think this is an example. What I expect is that this is this phenomenon that there is this copy in evolution with a different twist. Yes, I think that the difference between C1 and MBL and vicalin and colectins, yes, as well as the lectin pathway recognize small ligands on the surface in a fluid environment. So I expect them to work quite differently, actually, that it's not this particular structure-driven process, but much more a different process. So I'm not completely So I would expect differences. And every time when things are, and that's also, otherwise there's also no point in it. Right? So it's more, that's more a philosophical, that doesn't. Anyhow, but I would expect it to be different. Yeah, so that, I think there's a fundamental difference between the two processes.
Any more questions? Any more questions for Piet? Okay, so I have one. So imagine you hadn't already solved all the structures to the complement system, and you were starting again after the resolution revolution. What weapon would you pick? Would you pick crystallography or EM?
Obviously, I would pick EM. No, of course, no. You want to. This is a fascinating, stimulating field, right? So it's no doubt I would do EM. No, I basically tell my people in the lab there are two things that you can, which I would do if I would just graduate and move on. It's either doing neurobiology or EM. Now, since EM is already growing and happening, so the bigger questions are neurobiology. But no, I think it's in terms of structures. I think also the fact of putting this whole thing. What I'm really excited about is this physical work, to put it on the surface to actually see it working, because that obviously is very tricky in structure biology. How do you, and this now with the tomography, you can actually look at these things. It's still not reliable in terms of getting high numbers is a huge amount of work. Yeah, but still you can actually, it is clear that one could principally do that. Yeah, build up these complexes and study these things on the surface. And so already with the antibodies, it was a very unexpected result. And so I cannot see, well, yeah, that's not true, because the other, basically, by the fact that the crystal of the IgG had this hexameric arrangement, yeah, but that's a structure from 2002, I believe. And so it's, no, it's just data, of course, yeah. And so, no, EM is the way to go. No worries. But then, of course, crystallography has been great, because if we would have, yeah, because the fact that you sort out one conformation by crystal letters, yeah, so it's one conformation out of perhaps a bush of many, but obviously it's good to have something perfectly. Yeah, also the structure I showed with the overlay. Yeah, so all of these low resolution, high mobility things is a big problem in EM. Yeah, because it's these large motions of the same dome. So in that sense, crystallography is beautiful, because it just gives you a single conformation.
Yes, yeah? I have a couple of questions. So I was unsure what that plume, was that antibodies?
No,
you
that's...
had that sort of plume structure sticking off the membrane surface?
No, so in that particular case, we just added, started off with C5b6 and 7.
Yeah, that thing.
Yeah, so there's no antibody around. So those are, basically these kind of structures, C5b with 6 and 7. And they're just sitting here, and possibly you have like many, so what I expect is that you have actually many C5b6, 7, 8 complexes, for example, forming together a pore. Yeah, so all these plumes, this is basically the size of what you would expect. You take C5b6, 7 from the pore, then this size is about the same size, right, middle length. So these are the pieces of structure. Yeah, so this height, so here, for example, so there's multiple pores here, but here you have, for example, sometimes you see this, depending on where you are, but you see this kind of tower on this structure with the C5b, and that's basically the length that we see of these plumes. Right. So you have, instead of having C9 making the pore, you have C8, multiple C8s that perforate, and that gives you these smaller pores of 30 angstroms. And without C8, you just don't penetrate the membrane.
So do you think you could use like nanodiscs or something, these membrane substitutes to isolate these various membrane complexes? I mean, you could get more. So what Dorian did is actually, not with nanodiscs, but just because this pore itself, so the MAC pore, of course, is very, because of the beta beryl, it's very robust. Yeah, so you see actually purified from these vesicles, these pores, and they stay intact, and they do single particle averaging. No, but I mean, there's earlier intermediates where you have all those other structures.
Yeah. So heterogeneity is, I think, a problem, but it's also a force in nature, right? Yeah. So it's yeah. I don't have any reason to actually go and spend a lot of time in seeing exactly what this is. It's like there are other questions that before this. Yeah, so I'll tell you one question, which is an important question. This C5b6 has no thioester. Yeah, so cleaving it off makes it a soluble protein. So why? That's you have to target the membrane. Yeah, so how is this happening? Yeah, there has to be a process that actually you hit the membrane, otherwise it's completely waste. Yeah, so what people think is that the classical description in the book is, yeah, this is a soluble and goes off in solution, and then attacks this membrane again. I find it very unlikely. Right, so I would be interested in that question to figure out how this leads up to the step before. And we don't know this step either, like from convertase. What makes the shift of substrate specificity? And the ideas that are out there in textbooks, some of them are bizarre, I would say.
So those kind of interlocking MAC pores that you see, do you think that kind of interaction of multiple different MAC pores, do you think that's specific, or is that just a number of pores interacting rapidly next to each other and kind of becoming stuck together in a sense, or do you believe it to be specific in nature?
I missed the first bit of your...
So the kind of interlocking MAC pores, the multiple MAC pores forming these interlocking kind of figure-of-8 structures, do you believe that to be specific in nature, or is that a kind of just rapid interaction of pores stuck side by side?
So what I didn't point out, and I should have pointed out, of course, because the work here was done on this reconstituted system by starting with human serum. Here you also have these multiple pores. Yeah, so putting that together with these multiple C8 pores is, I think, that the convertase complex actually generates multiple starts. And they just make these, sometimes one and then it just makes C9, but sometimes you have like multiple C5b6, 7, 8 complexes together. And that just then doesn't do anything, and then you have C9, and that just makes the whole. So we also don't see any pores from half structures. We only see complete structures. And that's a big issue in the field. It's like, do you have, so this from the bacterial work, I think it much depends on the type of membrane. You get these hemipores and this toroidal structure of lipids. I think in mammalian cells these things don't happen. It's just energetically not favorable. Even in these vesicles it doesn't seem to happen. And what you just have is just first making up this whole ring structure that's much more stable. So I do think from that, and sometimes you just make complexes and they just don't end up in the membrane. Then they shoot up in solution. And that was in one of these movies. So I think you actually generate multiple of these complexes in one enzyme complex, the C5 convertase. Yeah, that's just why if it happens to have multiple, it hits first more complex, it just makes more complexes, making more aggregated structures, and some actually hit the membrane and others just fly off, and it's like everything happens.
Okay, we're in danger of being late for the bus. Well, very late for the bus. But please join me once again in thanking Piet and also Azra and all our speakers today. It's been a wonderful day.